why don't we all take about maybe 30 seconds to get settled in and then we'll get started. So good evening. Thank you so much for coming together for this special evening. For those of you who don't know me, my name is Vance Jackson, and I'm an Associate Professor of Counseling Psychology and the Director of the Dimensions of Nature course here at Green Mountain College. And I'd like to welcome you all to our annual Dimensions of Nature and Sophomore Speaker. Before I introduce our amazing guest this evening, I'd like to make a special announcement. The Association for the Advancement of Sustainability in Higher Education, or ASHE, is an organization that works to empower higher education faculty, administrators, staff, and students to be effective change agents and drivers of this program. Is the most widely recognized framework in the world for publicly reporting comprehensive information related to college or university's sustainability performance. On this Earth Fair Day, I'm pleased to announce that Green Mountain College has been recognized as the top baccalaureate performer by ASHE. GMC's STARS score of 81.82 is the highest score for any baccalaureate institution ever. In addition to the top baccalaureate score, ASHE awarded Green Mountain College the first ever perfect score for sustainability-based curriculum. GMC also attained perfect scores in the areas of sustainability research and air and climate operations. So I'd like to take a moment to just say a sincere thank you to all of us who work to make this green place such an extraordinary place to be. Thank you all. Now it's my sincere pleasure to welcome our honored guest to the stage. Please join me in welcoming Dr. Naomi Levine. Dr. Levine is a Gabilan Assistant Professor of Biological and Earth Sciences at the University of Southern California. Dr. Levine received her BA in Geosciences from Princeton University and her PhD in Chemical Oceanography from the MIT and WHOI Oceanographic Institute's Joint Program. Her graduate work focused on combining numerical models with observations leading to new insights into carbon and sulfur cycling in the oceans. Her postdoctoral work as a NOAA Climate and Global Change Postdoctoral Fellow at Harvard University focused on developing innovative mathematical modeling to explore interactions between climates and ecosystems. In 2013, she started her own lab at USC where she holds joint appointments in marine and environmental biology, molecular and computational biology, and earth sciences. Through the combination of biological, chemical, and physical observations with mathematical ecosystem models, Naomi's research examines the underlying mechanisms responsible for biogeochemical cycling in marine and microbial ecosystems, as well as climate ecosystem interactions and feedback loops. Her talk tonight, titled Small But Mighty, the role that marine microbes play in mediating our climate, will focus on developing a mechanistic understanding of how marine microbes respond to environmental fluctuations and how these individual scale responses can result in large-scale ecosystem shifts, as well as ecosystem climate feedback loops. She'll also discuss the complex interactions between climate and biology, as well as the interdisciplinary approaches used to examine these interactions. So please, let's give a great, hearty welcome to Dr. Naomi Levine.
Thank you, Vance, and thank you. I've had a really wonderful time here today, meeting with students and faculty, and really enjoyed myself and learning about some of the really great programs that you have here. It's a really special place. Let's give you a great introduction. I promise that this talk will use, explain some of those large words that I strung together. I apologize for that abstract. So what I wanted to do today is to take you on a journey into the oceans. And to let you know a little bit about some of the work that we're doing in my group at the University of Southern California, but also about climate science in general, and where the uncertainties are, and and where some of the new exciting advances are. So to just start at the beginning, we are currently in a changing world. And as scientists, we're observing that world change in very quantifiable ways. So this is a plot of data that I really like. I'm going to show 3 different versions of it. This is atmospheric CO2 concentrations over time. These are the seasons going around the circle. And you can see CO2 going up over time. You can measure that with a high degree of precision. Carbon dioxide is a greenhouse gas, so it traps incoming solar radiation and warms the surface of the Earth, and that's leading to warming surface temperatures, which we can also measure quite accurately. So these are, again, measurements of surface temperatures, the year is shown in the middle, over the seasons, and you can see each year is subsequently warmer and warmer and warmer. So we can observe increasing atmospheric CO2, we can observe warming climate. And those 2 physical changes are leading to changes in the Earth system. So one of these changes, which is also very observable using satellite data, is a change in the Arctic sea ice volume. And again, these are months going around, and this is percent change. So you can see the circles are getting smaller and smaller. So the amount of sea ice extent is retreating each year. So each year we have less and less Arctic sea ice. So these are 3 very physical dynamics which we can observe. We have ways to measure them and quantify them very accurately. These increasing CO2 and warming is leading to variable and more extreme events that we can also observe and quantify. So things like hurricanes. We have a lot of big hurricanes this year. This is the forest fire. We have the biggest forest fire in California history this year, the Thomas Fire. We've got more drought conditions. California just came out of a 5-year drought and more intense floodings. And so again, these are things that we can quantify. As scientists, we can go out and observe and measure their occurrences. And so there's a lot of data on this graph. The takeaway is that you've got time here on the x-axis, and this is the occurrence of frequency of hurricanes. And this is the trend line, and it's going up over the past about 20 years. This is the occurrence and severity of drought in western U.S. And the labels here are mild, moderate, severe, and extreme. And you can see we're getting more and more severe droughts over time. So again, these are quantifiable changes that we've observed over the past several decades. So the question that I focus on, and a lot of other climate scientists focus on, is where are we going? Where are we going to be in 2050, 2100, even if we want to think of 2200? What is the Earth system going to look like if we continue our current behavior, or even if we modify our behavior? And so in order to answer that, we turn to numerical models of the Earth system. And so this is the output of these type of models. This is what we get out of the models. So again, this is time running from 1990 to 2050 here. And this is your temperature anomaly, so how much warming we're seeing. And so that relates to that first circle diagram that I showed you. So you can see increasing temperature. The black line are the observations. And these squiggles are model predictions, numerical model predictions of the Earth system that are used to project climate change. And each color is a different scenario, so mainly based on human activity, how much fossil fuel we'll burn, increasing CO2, and then other sequestration and mediation techniques that can be applied. And so you can see there's a lot of different squiggly lines, there's a whole bunch of different models from 30 to 40 models that are run, and we've got a wide range of predictions. There's a huge amount of uncertainty in the predictions of how the climate system is going to respond to this increasing CO2. But they're all going up. There's always warming occurring in all of the different scenarios. And these are very complicated models. So these are models that couple the dynamics of the atmospheric system. So these are similar models to what we use for weather prediction. They're run globally. And coupling that together with ocean dynamics, so circulation, the Gulf Stream, and other major current systems that we know about, movement of water throughout the oceans. Ice, which is really important for circulation in the ocean, so there's sea ice model, and then a land model with trees that photosynthesize and sequester carbon. So all of those models are combined together, basically a 3-dimensional Rubik's Cube, millions of lines of code, and it takes the fastest supercomputers that we have months to run these simulations. So a simulation that goes out to 2100 is about a month to run on the fastest supercomputer we have. So there are not very many of these models in the world. There's only about 20 of them. And so the predictions that we have, there's a lot of effort that goes into them. and there's a lot of mechanism, we put a lot of mechanism into them. However, they've been primarily focused on physical changes, so those things that we have observations for, the temperature changes, the CO2 changes, sea level change, ice extent. How ecosystems and biology are going to respond to these physical changes is still an enormous unknown. We don't understand the implications of these future climate scenarios for the organisms that are living on Earth. So as a oceanographer, what I really care about is the oceans and the organisms living in the oceans. So for the rest of the talk, I'm going to just be focused on the oceans, though I have, as I mentioned, studied terrestrial systems, particularly the Amazon rainforest, so happy to talk about that if anyone has questions. So the oceans, we live on the blue planet. Oceans make up the majority of the surface area of the Earth. Oceans are responsible and a critical component of the global carbon cycle. So carbon is in the atmosphere, it's on land in the form of organic carbon in plants and in soils, and in the ocean where it's in the form of both dissolved inorganics, so CO2 dissolved in the water, just like in a Coke bottle, and in organic matter, so organisms in the ocean. Our current estimate is that oceans are responsible for taking up about 40% of the carbon dioxide that we as humans have emitted to the atmosphere. So almost half of all of the CO2 that fossil fuels that we burn, that CO2 has gone into the ocean, and most of this is a physical process, a solubility where the oceans dissolve the CO2 and it's sequestered into the oceans because the oceans are so big and can hold a lot of carbon dioxide. This is leading to ocean acidification, which is one of the key issues in active areas of research for oceanographers, though I'm not going to touch upon that today. About half of the oxygen production, so half of the oxygen that we breathe is actually coming from the oceans. We have to thank the oceans for also mediating our climate through carbon cycling and for oxygen production. And this is coming from microscopic photosynthetic organisms. So these are in general single-celled organisms that are photosynthesizing, just like trees do here on land. And they're making a huge amount of oxygen. And so really critical for our climate. So why care about these processes? Well, as hopefully most of you in the room know, we really care about greenhouse gases because changes in carbon cycling, so for example, changes in this 40% number could mean the difference between Venus and Earth. I think the biggest difference between Venus and Earth, they're about the same size as the atmosphere. Venus is a primarily carbon dioxide, high greenhouse gas atmosphere with average surface temperatures of over 400 degrees Celsius. So not a place we want to live. All right, so what are these organisms that are responsible for the oxygen production and play an active role in ocean carbon cycling? So if we look in a single drop of water in the ocean, there's a huge amount of diversity of these single-celled organisms. And for the artists out there, they're really beautiful and super cool. So this is a picture of a whole bunch of different diatoms. They have all these different morphologies. They form chains. They've got weird spiky things. We've got coccolithophores that have these armored shells on them. So they're amazing organisms. And this type of diversity we've known about for quite a while. You can look under a microscope and see it. But if you take a drop of seawater out of the ocean and you look at it under a microscope, most of what you're going to see looks like this. So pretty boring. These are single-cell bacteria, heterotopia bacteria in general. Some are cyanobacteria. They've been stained so you can see them. It looks like a starry sky. They're just little round blobs, and they sit there and they don't look like they do much. So for a long time, we didn't know what the majority of these organisms were doing. We didn't know who they were. We didn't have a morphology, a way to identify them like we did for those really beautiful organisms. We didn't know the role that they were playing. So with the advent of the molecular revolution, so this is the ability to sequence DNA and RNA and look at the genes of these organisms, we now have a much better sense of what this diversity is in the ocean. So we know that there's about 10 to the 6 cells per mil, so in 1 milliliter of seawater, it's about 1 million cells. And there's an immense diversity of organisms out there if we think about them in terms of different species, incredibly diverse. This is what we call taxonomic breakdown. So if you look at all of different organisms that are in the ocean, most of them are bacteria. This green bit is bacteria. And we've got a little bit of viruses, some eukaryotes, so those are like us, but single cells. And then a lot of things we don't know what they are still. What I find really exciting and motivating is this little graph here. So this is from a pretty recent study where oceanographers went out on a sailboat around the world taking samples and sequencing everything that they could find at a bunch of different depths and different size fractions. And they compared the genes that they found in the ocean to the genes in one of the most studied microbiological systems, which is the human gut. So in the human gut, we know about 90% of the genes. So that's what's shown here, right? So if we look at the genes that are there, we know about what 90% of them are doing. In the ocean, we don't know about 40%. We have no idea what they're coding for, what their function is, why they have them. And what's really cool about this, or the science is exciting, is these organisms are living in a desert, pretty much. They're what we call really streamlined. They don't carry a lot of extra baggage. If you're going on a long, long hiking trip, you're not going to carry a lot of extra food just in the off chance you're going to need it. You're going to really think about what you need and be very careful about the weight that you're carrying. These organisms are doing the same thing with their genes. They don't carry around a lot of extra genes in the off chance they might need them. They only carry around genes that we think that they need to use. So our best guess is that these are really important and we have no idea what they are. Okay, so what do we know? So we know that there's a really dynamic world out there. So this is an illustration from Roman Stocker. I'm sorry, I forgot the citation. It's one of my collaborators who's in Switzerland from graphic artists. And we know that there's a lot of really dynamic interactions that are occurring at the microscale, the scale of these individual single cells living in this very big ocean. So we've got single cells that are photosynthesizing, fixing organic carbon from CO2 and producing oxygen. There's heterotophic bacteria that like to swim around and interact with the compounds being produced by the phytoplankton. There's grazers or zooplankton that like to chomp down and eat them, both phytoplankton and bacteria. There's sinking particles, and those are usually poop, fecal pellets, coming from zooplankton or fish or other things that sink because it's water. They're more dense and they'll sink down. And so there's a lot of really dynamic and exciting ecological interactions that are happening at very, very small scales. To kind of put in a little bit of a bigger picture, why would we care about these single-celled organisms? So these are the photosynthesizers. There's the ones producing the oxygen. They are the base of the food chain in the marine system, right? So these are the grass or the trees comparable on land. So if we care about things that we do care about as humans, like fish that we like to eat, or charismatic megafauna, as we like to call it, dolphins, whales, and other cute things to look at, we need to understand what's going to happen to their food source, what's at the base of the ecosystem. So there's two main motivations to the type of work that I and my colleagues do, and that in terms of understanding how these microbes, these single-celled organisms, both the photosynthetic ones and the ones that consume organic carbon, how they're going to respond to changes in climate. And the first is climate feedbacks and interactions with climate through changes in carbon cycling, so more or less photosynthesis, for example. And the second is ecosystem shifts, how changes in who's there is going to impact what type of fish can exist in the abundance of fisheries. So climate feedbacks is one of those buzzwords or terms that the scientists like to use. What do we mean by that? Well, so climate feedback is when you change some property of the climate system. So for example, atmospheric CO2, that changes the climate, warms it, for example, that changes something about the climate physics, maybe changing ocean circulation, that further changes that initial feature that you perturbed. So an example is, we're putting atmospheric CO2 into the system. That's creating warming. That's changing ocean circulation, which is actually releasing additional CO2 for causing less uptake of CO2. It's also changing biology potentially through less photosynthesis and more respiration, which is also increasing CO2. This is what's called a positive feedback. You can think about it as a runaway cycle, right? One small change in this, you keep increasing, 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 increasing. There's also negative feedbacks, where you increase this and it gets dampened by a process further down. So it's these positive and negative feedbacks that are leading to a lot of the uncertainty in our climate predictions. The Earth systems are very, very complicated system with a lot of interactions, and some of them are positive and some of them are negative. And so predicting these interactions and their magnitude becomes challenging and also really critical. And so what I study is this, right? Which, unlike this arrow might imply, looks a lot more like that. So it's very complicated, the interaction between physics and biology, and how that might result in a climate feedback, so a further change in the climate system. So another way to illustrate this is through this diagram. So when we think about the ocean system, it's incredibly complicated in terms of the microbes that are there and diverse. So that's my little schematic of microbes in the ocean. They're all interacting with each other, and this is a statistical analysis, a hairball plot, basically, we call it, where each little dot color is representing maybe an organism or a temperature or nitrate or some other process we can measure, and all of the lines are illustrating the connections between them. So we can, we know a lot about the diversity and how they're interacting. These interactions are also coupled to a really dynamic system, which is varying in time and space. So there's a lot of storms in the ocean. It's very patchy. This is a satellite image. This is a model, but satellite images look similar. Where you can see all of these pretty dynamics, like if you have a coffee cup and you stir in the milk, those pretty swirls and patterns, we see that in the ocean. So those physical dynamics are interacting with a very complex biological system. But when we try to model this system, we can't model this level of complexity. And so we have to simplify the system down quite a bit. And so we go from a picture that looks like this to one that looks more like this, where the physical environment is a lot less patchy and dynamic. We have a lot more smooth and constant physical drivers, changes in temperature, for example. And we have a very simplified ecosystem and very simplified interactions between the organisms that live in the ocean. And so our current set of numerical models that we use for climate predictions are using a model framework that looks something like this. And so they're limited in their ability to understand these implications for future climate systems, because they are not resolving these really important and critical interactions that are happening at very, very small scales. So what we look at in my lab is how do we take what we know about small-scale diversity, these microbes that are living in the ocean, what function they're playing, or we hypothesize that they're playing, interactions between them, and go all the way up to the global scale and try to understand the role that these very, very small organisms might be playing in very large-scale processes like carbon cycling and climate change. So in order to do that, and ask this kind of primary question for my research of what the role of diversity is in climate cycling, I combine a bunch of different interdisciplinary techniques. So we use theory, so building on a strong foundation of theory and both ecological theory and physical theories. We use in situ observation, so that's actually measurements that have been taken by oceanographers going out into the field, taking up water and measuring properties such as who's there, the chemical, the physical properties of the water temperature, salinity. And we combine that with numerical models. So those are computer models where we create mathematical equations to represent what we think is going on in the system, and we can run those forward in time. So I'm going to talk about two little vignettes of studies that we're doing in my lab that tries to address this question using these interdisciplinary observations and what we found. So the first question we wanted to ask is this diversity even important when we think about climate change? So climate change is happening on a very, very large scale, the scale of our planet, right? The whole Earth is warming. How important is it that there's lots of different types of bacteria, single-celled organisms in the ocean? Maybe for questions of climate, that's not relevant. Maybe we can ignore that diversity and we don't have to worry about it. And all we need to worry about is larger scale processes like the Gulf Stream or larger scale physical dynamics. Or maybe these single-celled organisms that are so small are contributing significantly, and by not having them in these models, we're actually underestimating or overestimating the potential warming in the system. So how much is diversity, or the lack of diversity in these models, contributing to uncertainty in our predictions? So to do that, we focus on a very small question, and that is it important that photosynthetic microorganisms are living together with what we call heterotrophic organisms? So photosynthetic organisms are the ones that can fix CO2, so they're like the microscopic plants in the ocean. The heterotrophic ones are the ones that eat organic carbon and produce CO2, consume oxygen usually, and produce CO2. And in the ocean, they're living together. If you remember back to this plot, they're in close proximity to each other. The numerical models that we use for climate change don't have these guys in them. So is that an oversight? Is that a simplification that we're comfortable making, or is it one that could be biasing our results? So to do that, we used a data set that my collaborators at the University of Southern California are collecting at the what we call SPOT site, San Pedro Ocean Time Series. So this is LA here. This is Gabilan Island, which is a beautiful place if you're ever been to LA. And in the middle between the two, we have a site. So what's kind of funny about oceanographic sites is that they're just latitudes and longitudes that we randomly pick in the ocean. If you were to take a boat out here, you wouldn't notice it. It doesn't look any different. But we go back to that same latitude and longitude every single month, and we've been going back there for 20 years, measuring everything that we know how to measure in the ocean. So the chemistry of the ocean, carbon, nitrogen, phosphorus, iron, the temperature, and then the organisms that are there. So we go out on boats. This is not from this site. We don't get waves that big. But we put in instruments that look something like this, if you can see it. So they're basically these cylinders which we drop over the edge, and we can collect water at different depths by closing the top and bottom at the discrete depths that we want to. And we bring that water back on the deck and we filter it. So most of oceanographic field research is filtering. It's taking water and putting it through a little paper filter, basically, and then you collect what's on the filter or what went through the filter and make your measurements. So by looking at these observations that we have over a 20-year period, here we use 11 years, the first middle 11 years, we can look at how measurements of these photosynthetic organisms and the heterotrophic organisms have changed over a season. So this is month now, January to December, and the bars are the heterotrophic bacteria for two different types of bacteria, the green type and the black bars, and then the photosynthetic ones. And so this is our spring blue. That's like the green up that we get on the land, the big burst of photosynthesis that happens in the spring time. And so that's the most photosynthetic production that we have here. And about a month after, we get a peak in bacteria, which are eating up all that yummy photosynthetic material. And this green one is really good at eating new organic carbon. The black guys are just kind of there all year round, and they're pretty good at living in the summertime when things become more like a desert and there's not a lot of food for them to eat. So we get this kind of oscillation in the heterotrophic bacterial community of the type of organisms that are there, that we can see in the observations. So once we saw that, and this is actually coming from genomic analyses and statistical correlations studies, we asked, okay, if we put these two types of bacteria in our model, what does that do for us? Does that help us understand carbon cycling at our site? So to do that, we built two models. So we started with a classic model. So this is a schematic of what a classic marine ecosystem model looks like. And so each box is a different component of the ecosystem. The green one is the photosynthetic guys. There's actually two boxes, one for little guys and one for big guys. This is the things that eat that one. So the carbon's running from the photosynthetic guys to the things that eat them. And we've got inorganic is the CO2, basically. So the arrows are how carbon is moving between different members of the ecosystem. The boxes are different members or organic carbon pools. So we took this standard model. This is the model that's in all of those big climate models in terms of ecosystems. And we added 2 bacteria groups. So that's this little pink box. So we took exactly the same code and we added some extra lines of equations to represent those 2 bacterial groups that oscillated. And we asked, does that change our system? So the first thing you do with a model is you try to validate it against observations. It's really important to make sure your model is at some level representing reality. And so we did that extensively, so we can look at observations, and we've got lots and lots of observations at this site, which is great. So we can really constrain our model. So we can plot out how much photosynthesizers are there versus the observations. This is a specific type of photosynthesizers, say in a bacteria. They've got slightly different pattern, and we can reproduce that. If we look at the bacteria, this was that figure I showed you previously, where you see that spring bloom and a lot of phytoplankton, and then you got that lag in the bacteria. It's not perfect. Models are never perfect. My graduate advisor told me if your model is perfect, you did something wrong, because your model is never perfect. You don't know everything. But we get that lag, so our green peaks a little bit after the yellow, and we've got these black guys that are just kind of hanging out and do better in the summer. And then we did an experiment with our models. So a lot of people don't realize the modelers do experiments too. We just do them with our numerical models. So we took our model and we did an experiment. And we said, what happens if we pretend we're 100 years in the future and we're in a warmer environment with less mixing in the ocean, which is what the projections are? How does that change for our 2 different models? And so we can do that in the model. So each of these lines is a model simulation. So the temperature is warming. So the black is the coldest, and we run a whole bunch of different scenarios where the temperature is warming in the model. And then we can look at what the 2 different versions of the model project. And the takeaway here is they project very different things. So they start out in exactly the same place. We validated against the observations, and they both did equally well. But when we run forward into the future, so future is going this way on the axis, and this is for winter, spring, summer, and fall. The original model, which is in blue with no bacteria, and the yellow model, the model with bacteria, which is shown in yellow, start to look very, very different. And that's because they're fundamentally different models with different mechanisms. So we've taken our knowledge about how the ecosystem works, and we've improved our model. And while we can't tell under present-day conditions that it's any better, we can see that it does have a very different prediction for what the future system is going to look like, suggesting that our current models that we project climate change with may have additional uncertainty in them. Here it's hard for us to project whether that's too little warming or too much warming, but now that we've identified that potential error in the model, we can now go in and ask what the impact is for a climate model. And so that's in part what we're doing. So we're running, these are simulations my postdoc has done, looking at global versions of this model. And as you change the bacteria type that are in the model, you can actually have very different photosynthesis. So that's the only takeaway from this, is that the color bar is the same, but the colors are different when you go from one bacterial group to a different bacterial group. So those single cells that look like the starry sky when we look at them under the microscope, which we didn't know 20 years ago who they were or what they were doing, actually could play a really big role in carbon cycling and really change how the oceans interact with the atmosphere and how much CO2 stays in the atmosphere, and therefore how much warming we're going to see here on Earth. And so, as I mentioned, one of the big motivations to my research is climate feedbacks. We haven't talked about climate feedbacks with this study, but one of the things we want to understand is as we move to a warmer and warmer world, will these interactions that we've identified as important create those climate feedbacks and move where these wiggly lines are projecting will go in terms of temperature. Another area that we're looking into is what happens to those microbes as you go forward in time. Right? If you could pull out a sample of water now and a sample of water in 2050, will those microbes look the same, or will they have evolved? Will they look differently? So these are single cells. They're dividing about once every 2 days-ish. So there's a lot of generation time, so a lot of potential for evolution to happen over this pretty short period of time. And there's a pretty big pressure for evolution because we're changing the climate so quickly. So I believe most of you in the class anyways read some of Darwin's work. This is the same process, right? So he was thinking about macroevolution. We're looking at single cells and whether or not these little green dots are going to change some of their genes and how they work in the interact with other organisms and maybe their growth rates and change to look more like these blue guys. So only recently have we been able to get some insight into this, in part because we can now actually look at the genomes, right? Because they're not going to change from green to blue, unfortunately. They're still going to look like single cells, but how they act will be different. But we can go in and sequence them now, and we can actually look at how their genes are differentially regulated. So there's been a couple of studies now, one of them at USC, and one in Germany, and one in Australia, where they've looked at evolution in a bottle. So they've taken a culture of these single-cell organisms. They grow them up in the lab. For ocean organisms, we grow them in liquid media, be more like the ocean. Some of you may have worked with plates. I saw some downstairs in the lab. It's the same concept. So we grow them in the lab under different conditions, and this one was done under different CO2. So they take the same organism, they put it under present-day carbon dioxide, and they put it under future carbon dioxide, so a doubling in this case. And then they can monitor them, and they've run this experiment now for 9 years. So that's a lot of generations that they can look over. And they can sequence them, and they can actually observe a genetic response in these microbes. So there's actually differences in the organisms that stayed at present-day conditions and the ones that got plopped into a future condition. So we now have the tools and the capability to test how organisms are going to evolve in the ocean, which is really exciting because unlike birds and other animals that have much longer generation times, it's very hard to test evolution. You can't wait long enough. Whereas here we can wait 700 generations in the matter of a single PhD and still get some answers. So what we're doing in my lab is taking this brand new research, this is all just actually coming out and being published now, and saying, can we use this to inform the models and understand the implications that this might have for carbon cycling and climate change in these large-scale models? So again, we try to couple the observations with the models, so we're able to take different measurements that were made at different points in time in this evolution in a bottle experiment, and we can take the model and we can make sure we can fit the model to the data. So these circles are data, the line is a model. I can go into the more details afterwards if someone wants to come up. But the main message is we're able to get the evolution model to match the observations. And then we're doing some really cool experiments with the model. So if we take an ocean model, we can throw tracers in like a little rubber ducky into the ocean. And we can track where it goes, how it moves, and what environments it experiences, and we can record them as they move. The little rubber ducky tracer moves around through the model. So that's what these little squiggly lines are showing. All the different rubber duckies we plopped into our model and we watch them move around. Then we can say, what happens if we actually had microbes in there, just like we did in the lab, but they were experiencing these different environments as they were floating around through the ocean, the different storms, moving into the tropics and out of the tropics, all these different variable environments that organisms in the ocean actually experience. What happens? When do they evolve? When can they evolve to new climates and adapt, or are they going to die out? So this is a study work that we're still doing. It's actually ongoing research in my lab. But we're finding some really exciting stuff where we can look at the trajectory as it's moving along, and all of a sudden evolution happens, and we hit upon the perfect organism that's ready for that environment, and it takes off. And so we see these sweeps, basically, where we get evolution happening. And it's happening on relevant timescales, so it's happening on quick enough timescales that we could potentially observe it in the ocean, but it's not happening instantaneously, and that's really exciting scientifically. So we can't assume that these organisms that are in the ocean are going to be perfectly adapted to our changing the system, which is what marine microbiologists have kind of assumed for a long time, that they mutate fast enough, they're going to evolve really quickly, we don't really have to worry about it. And what this work and the work of my collaborators who are doing these lab experiments are suggesting is maybe not. Maybe it's going to take them, they will evolve, but maybe it will take a decade or two for that evolution to happen. So a lag in that evolution, which has implications for the climate system. So those are just two of the studies that we're doing in my group, all focused on trying to understand this very big picture. So this is a satellite image of photosynthesis. So those are those photosynthetic organisms. You can see them from space, from these satellite images. It's a movie, and the green is an estimation of how much photosynthesis. So you can do the same thing for land, and you see that green up happening in the spring, and then the ice, also snow and ice moves back and forth. But the same thing is happening in the oceans, and you get these bloom events. You get lots of really cool dynamics happening. And so we're trying to understand this system and how we can better model it and better understand how oceans can play an important role in the climate. And so I wanted to end, I know as part of the class and this morning there were some questions about how I got to where I am. And I think that oftentimes when you look at different researchers, and even if you look at their CV, it looks like they've taken a linear path. But in reality, my path anyways, and I think a lot of our paths look a lot more like this. So if you're an undergrad or a grad student sitting here and you kind of have a sense of where you want to be, it's never a straight line between the 2 points. So as I mentioned, I started out as a hard rock geologist. I did some volcanology. I studied the composition of the interior of the Earth. So a field trip I took my freshman year out to California and Mammoth Mountain. I then worked for the Environmental Defense Fund, so an environmental nonprofit doing policy work, translating scientific research into briefs for senators to help advance environmental policy, which was really fulfilling and also convinced me I wanted to go to graduate school and become a scientist. I then went to graduate school and I spent about a year of my life on research vessels making measurements. I made chemical measurements, I did biological measurements. I did a whole bunch of modeling, computer modeling, on very different topics. I had a schizophrenic thesis on half on carbon, half on sulfur. I then did a postdoc in terrestrial ecology in the rainforest and how the rainforest was going to respond to climate. So these are a lot of different topics and a lot of different techniques and tools. And now I am studying variability in the ocean and timescales and impact of microbes in climate. So these seem like a lot of very different tangents, and in fact, at the time, it didn't feel like there was a common overarching thread. But when I look backwards at what I'm doing, there's actually a really nice linear progression where I can actually look at how I started with looking at respiration and heterotopia bacteria in the ocean. I was asking a slightly different question, but it was a similar idea. I was looking at genes and how they were regulated in concentrations and building computer models that address variability, which has all kind of come together and allowed me to have the perspective that I have today and do the kind of science that I'm doing today. So the message I wanted to leave you with, and we'll end so there's time for questions, is 2 things. First is that you never know where your path is going to take you. This is me and my husband in college. This is us this winter with our 2 little boys. There's a lot of balances and juggling, but if you're doing what you love, it makes it easy. And then listen to your gut and follow your passion. And never be afraid of a challenge, because the challenges are where the really exciting things happen in all different fields. So I wanted to end by thanking the funding sources. I've been really fortunate to have funding from the National Science Foundation, from the Sloan Foundation, the Simons Foundation, the Moore Foundation, University of Southern California, and then my lab members. So I've got an amazing group of people who work with me, undergraduates. I've got, had a whole bunch of undergraduates in the lab. I've got a whole bunch of graduate students who've done some really great work, lab technicians and some postdocs, so postgraduate researchers who are working with me, and all of this work and the work that we're doing is contributed by them. So with that, I'm happy to take any questions.
Thank you for that talk. And you might have covered this already. There's a lot of information, but what are your thoughts on the microbes' ability and other sea creatures that photosynthesize or use photosynthesis bacteria and stuff? What are your thoughts on their evolutionary process spiking in its compatibility with the new environment, and how are they? Do you think it's possible for them to change their increase in evolutionary rates for that?
That's a really great question. So the short answer is that we're not sure, but I think that there is definitely the potential for both the photosynthesizers, so those are the microscopic plants, and the bacteria that eat what the photosynthesizers produce, to evolve. And we see that in the lab. And so there are certain changes in climate, and particularly the CO2, they seem pretty good at evolving and adapting to. Temperature becomes a really challenging one. So if we look at temperature curves in the ocean, for organisms in the ocean, they all kind of fall off once they hit 34 degrees. We don't have a lot of organisms that can grow well past 34 degrees. Now there are organisms that grow in hot springs and in these really hyper environments. So it's possible for life to do that, but it's a really interesting phenomenon where we see temperature adaptation at the high end doesn't seem to exist in the current ocean. So the question is, and I think this is a really big unknown and we don't yet know, is that just because they don't really see it enough to have ever evolved, so we don't see organisms that can do it because the temperatures are very rarely above 34 degrees for a long period of time, or 35 degrees Celsius, or is there some sort of enzymatic process? So enzymes start breaking down and the cells just can't function in the way that they need to in the ocean at those higher temperatures. And so I guess the question is, the answer to your question is, we think that they should be able to evolve. We're not 100% sure they'll be able to evolve to all aspects of changes in climate.
Others? Dolphy?
My question is, have the researchers at your lab been able to, or find it helpful to look into the past at various climatic changes to see if there's a relationship between the biota in the ocean and the climate?
That's a really, really fantastic question. So, so past climates are incredibly useful for trying to understand current improving our current predictions. And there's a lot of paleoceanographic work that's being done to try to constrain primarily actually what the past climates looked like even. So for example, how ocean circulation patterns, where water movement happened and where ice was and wasn't. That is really challenging in itself. There are some work that can look at some changes. The problem is DNA doesn't preserve all that well over long periods of time. And so we can't get at these cells that don't have protection thresholds. So some of those really beautiful images that I showed, a lot of those have shells that are made of silica, so that's what forms opal, or calcium carbonate, the white cliffs of Dover, those are fossilized versions of those. So we can look at shifts in those types of organisms and changes in their morphology over time. But it's harder to get at the DNA. There are some labs that are working on sequencing paleo DNA. The question is how good it can be at constraining diversity changes. But the other challenge is the timescales involved. So while we do have good analogies for the magnitude of the change, so the increases in CO2, for example, if you go back to the Jurassic transition, there's a huge change in CO2, but we think that it occurred over a lot longer period of time than we're currently foreseeing the system. So the evolution timescales would also be very different. So it's a challenge, and there's a lot of valuable information to be gained from those studies, but unfortunately I don't think they're going to be the whole way to answer the problem.
I got one back here.
Yeah.
In the increase in climate change with temperature and increased carbon levels, do you see more of a difference in seasonal ocean environments, or would you see them more on the equatorial side?
That's another good question. So, unlike land, where we can easily measure occurrence of hurricanes, for example, it's harder to measure and predict variability in the ocean at the scales that are really important for organisms. So we don't really know how the oceans are going to change. We have an idea of how their overall patterns are going to change. So when you warm the surface of the ocean, it stratifies more. So if you think about a lake in summertime, it's warm at the top, and then once you dive in, it's cold, right? So that's a stratification due to the warming of the surface. In the winter, right before ice is over, right, the temperature, everything's mixing up more, so it's less stratified. So as you warm the climate, the ocean system is going to stratify. And so there's kind of a transition region. So the middles of the ocean dryers right now are classified as the deserts of the ocean. Right, so they're dark blue here, there's not much living there. They're already pretty warm and pretty stratified. They're going to get more warm and more stratified. These areas in the poles are going to be the winners of the situation. So the reason why there's black here is the cloud cover problems, light. But the highest productivity is actually in this kind of band here. So there's some areas in the poles closer to the Antarctic and Arctic regions where warming is going to be a benefit because there'll be more light available for the organisms. And so I think that the biggest uncertainty is in the intermediate ranges where we don't really know when they're going to get pushed over into being too warm and too stratified.
Other questions?
I'd like to recognize you put a lot of research into everything you do, and I'm wondering how you determine what you'd like to research more and get funding for. If you've got a project coming up, how do you know it's one that you want to invest a very long amount of time into?
Yeah, so a couple of things. I think my challenge is getting excited about too many things and wanting to do too many things and not, my goal is to focus more. So I think we do a lot of preliminary work before we actually submit grant proposals to write to get new funding. So for example, I've had someone working for me for the past 6 months trying to figure out if we can use statistical approaches to understand some of these big data sets that we get from the ocean, to then improve the models in a specific way, which is, I think, a pretty high-risk proposal, and could be one where if I had written it 6 months ago, could have ended up in a year regretting having gone down that path. So in this case, I had him work on it, not funded, called bootstrapping on other projects and not my startup funds, to get some preliminary data that convinces me that it's something I really want to work on and I'm really excited about still, and then also allows me to convince funding agencies that this is something really cool and will work. Because that's what they're really looking for is it interesting, is it going to make a difference, and do you think it's going to work? Is there a high chance that it will actually produce some interesting results? But the thing I love about oceanography is it's incredibly interdisciplinary, not only my work, but most of the field is very interdisciplinary. I think it harkens back to the fact that to actually go out and sample, we have to go out on these research boats that cost about $30,000 a day to run, probably more, because that was the number that I had to deal with in graduate school when I was dealing with the boats. And they move at about the speed of a bicycle, so they don't move very fast. And so in order to get places in the ocean, it takes a really long time and it's really, really expensive. And so we have to come together collaboratively. And so we've got physical oceanographers, biological oceanographers, chemical oceanographers, marine geologists, all working together on projects, interdisciplinary projects. And so that is one additional factor that kind of factors into putting these projects together, is how do you find interdisciplinary and exciting work, and how do you bring a bunch of cool people together to work on them.
When you get the results, is there somebody that you will send it to, so that, like, somebody who takes action on, like, results that you will get? I don't know.
Yeah, so like policy related. Are you thinking about policies?
Yeah.
Well,
no. More. Yeah, that the people that, like, I know that you guys get all the results, but is there somebody who takes action on it, like policy change, but also, yeah, I guess policy. Yeah, policy change.
Kind of. So when we get our results, we write them up in journals as articles, and there's specialty journals, which are probably only read by people like me in my very small field. And then there's more general journals, so Science and Nature, which are read hopefully by not only scientists, but other science literate people who are excited about learning about new cutting edge science. So we write them up in scientific journals. And that is, I then kind of step back from the process, and what happens is usually either NGOs, environmental nonprofits, or government agencies step in and synthesize all of the knowledge that are pertinent to whatever policy issue they want to address. So sea level rise, right? They'll go out and look at all the published studies on sea level rise, and then come up with what the current scientific understanding is on where sea level rise is going to be the greatest, and then generate a policy or law or based on that. And then the scientists come back in usually. So I haven't done it yet because usually you have more gray hair than I do. But my graduate advisor, for example, goes to Washington, D.C. a lot and testifies to Congress in support of policy measures. So typically the scientists are not involved in writing the policy, but we can support the policy by coming in and explaining our science in hopefully ways that policymakers can understand, and explain how we've come to the conclusions that such and such is going to happen.
Other questions?
So one of the discussions that we've been having in my class is about what it means to be a woman in science, and what it means to bridge the sometimes some stereotypes and the social environment that comes along with being a woman in a STEM field. And I would love to hear you speak to that, and to challenges that you face, and things that have helped you overcome that, and role models.
So it's a really important issue, and something I'm really passionate about, and trying to support women scientists. So I think there's a lot of different facets to that question. I think that there's a cultural change that needs to happen, and that needs to not only be women scientists changing it, but male scientists also need to show that they can support women. So I have a colleague, for example, who is the primary child care provider for his family, and I keep telling him, you need to make sure your students know this, right? Know that it's not just the women who have to be in charge of child care, that you're the primary child care provider. And so I think that recognizing that not only having more women in faculty positions, but also having men that are actively showing what it means to be an equal partner is really important. I think that increasing science education to K-12 is actually really, really critical, and getting girls really excited starting out when maybe before they get to college. What I found really, really supportive, especially at the faculty level, is finding a group that can give you a voice. So at USC and a lot of other research universities, usually it's when you have a lot of science and engineering faculty, because we've got a pretty big group of female faculty members. There's Women in Science and Engineering, or WHOI programs. This was actually a really big driver for me going to USC, so there was a huge endowment of $20 million to the WHOI program at USC. And more importantly, they run it incredibly well. So there's a faculty member who's the director, and she listens to what's going to help the women in science. So opposed to deciding that X program is going to be what they really need, she has focus groups and try to understand what do you actually need. So they've implemented policies like a travel fund for young mothers. So when your kid's younger than 5 years old, you can apply for funds to bring the child with you to conferences, or a child caregiver, or bring a child caregiver to your house while you leave. To really have acknowledgement of the fact that going to academic research conferences is where ideas happen. So most of my projects are coming out of collaborations and ideas that have come out of research conferences, and if I had to take off 3 years from traveling to conferences because I had young kids, I would be falling behind a lot. And so in having someone that I can turn to when they closed the child care center at USC, and I had no child care, and I could go to them and say, this is a problem. We need child care for our women faculty members. We can't work if we don't have child care. And they can go to the provost and say, fix this. Has been really advantageous so you don't feel alone, I guess is what I'm trying to say. Is trying to figure out a way to not feel alone in it, because you're not alone. There's a lot of other women. And also not trying to find role models and realizing that role models don't have to look exactly like you. And I think that I don't know how to convey that, but it's going to be impossible to find a role model who looks exactly like you do. And I stopped going to panels on work-life balance and women in science for a long time because I kept sitting there getting more and more depressed and frustrated because I never saw myself on the stage. They never had partners that did the kind of work that my husband does and the challenges that we face. And I finally realized that I was never going to see myself up there. But what I had to realize was that other people could deal with their own challenges, and there are strategies to deal with the challenge. And you just need to find the help and support that you need and ask for the mentorship. I think that oftentimes everyone gets busy, but we all want to be good mentors. And I would say the majority of both female and male scientists want to support young scientists. But sometimes we forget, or more, like the time goes by so quickly, sometimes I forget that I haven't talked to somebody in a month, not because I didn't need to, but it just, I can't believe a month has gone by. So asking for mentorship when you feel like you need it is sometimes hard, but is also really important.
Other questions?
So you had mentioned that funding largely influences the topics of research that you do. Would you say that is true? And if so, is that limiting to your research? And are there topics of research within your field that you feel like you wish you could find funding for and are valuable, but isn't possible in the situation that we're in?
Yeah, so I personally won't work on something I'm not excited about. So I won't apply for funding for a project I don't want to work on just because there's funding there. And I think that most scientists feel that way. That there is funding to be had. And sometimes you have to do maybe the less creative cutting edge science in combination with the science that you really want to do, right? So, but I've been very, very fortunate in not having to limit my science based on funding. So I've been able to find the funding agencies that really support the science I wanted to do. And a lot of that comes down to communication, I think. So, and luck. But being able to articulate and clearly show, demonstrate that your ideas are exciting and new and interesting, something you want to spend money on, and identifying who you're pitching that to are really important. but I think it just depends on the situation. So there's a lot of researchers who are not at universities who struggle more with it because they have to raise more money. So I'm lucky because I'm at a university. So I have money for me, my students have money through teaching fellowships, or if I didn't have a grant to pay them. So I just need a little bit of money to get my research done, opposed to someone who has to support themselves and all of their lab members and the research. That's a bigger grant you have to get funded. So you have fewer projects you can actually accomplish because you need more money to do it. So it's the difference between a research university, that's what we call soft money, or just research and a university setting.
Other questions?
Hi. I'm curious if you know of any research into the impact of plastic buildup in the ocean on these microbe populations, and if it has any impact.
So there's just some new studies that are coming out. I think you can speak on there are microbes that are able to degrade plastics in the ocean. It's endogenous resin. These microbes are incredibly versatile and consume pretty much anything you give them. The question is how fast they can grow. So I do not think that microbes will be the answer to plastics in the ocean. I think that the organisms that can consume the plastic are not going to grow fast enough to actually make any serious dent in plastic presence. And plastics are just hydrocarbons, so if they've got the genes to degrade them, there is not an inherent negative to them eating the plastics. The negative comes into if there are metals or other toxic compounds that are co-occurring in the plastics that could be negative. Usually it's not a negative to the bacteria, especially in the ocean because it's a fluid environment, but it could be negative to a negative factor for things, higher level organisms like fish and other things. Again, probably not in the open ocean environment, but potentially in estuarine systems or coastal systems where you kind of more closed system, things can build up faster. So we know that hormones, for example, that are coming out of wastewater discharges from actually usually birth control pills are significant enough that we can measure them and have an impact on marine life in those regions.
Any other questions?
Thank you for coming. My question is about access to this kind of information. To this kind of information, your research and other science like this. So we're lucky to obviously be in college and have this as a resource, but what about for people who don't have college or don't have those newspapers or magazines? How do we get this information across? Because it seems like very important information for the future of this planet. How do we include every voice within this conversation?
Yeah, that's something I struggle with a lot. And I think it's actually an even more basic problem is how do you increase science literacy in our population in general, because I think that that's really critical to increasing or decreasing the polarization in our society. I don't have a good answer. I think that we just need to do a better job on education funding, and while I would love everyone to understand what I do and be able to get out there, I don't think that that's the first order problem. I think the first order problem is just basic logic and science literacy in school systems. And so I do a lot of outreach in the local LA area, but it often I'm personally, but it's good, and I'm really glad that I can do that, but it's not those communities that need it the most. It's the communities that are really far from universities, and I don't have a good way to get there, and I'm not sure what the solution is.
Any other questions? Okay, well, thank you so very, very much for coming.